The invitation. As Mario told us, TMS EEG is indeed a powerful technique, and our aim in our lab is to, together with collaborators, is to develop real-time algorithmic closed-loop stimulation with multi-locus TMS. And here's an artist's view of what it might look in a few years. And so the idea is to perturb the brain with multi-coil TMS, with which you can electronically decide where to stimulate. And now, instead of a human operator in this closed loop, there would be a computer doing an algorithmic decision where to stimulate based on EEG. So EEG is essential here, and it's important that we get this done right. And I'm grateful to this discussion that we can improve. So we're going to do this future multi-coil algorithmically controlled TMS in a ERC synergy project called Connect to Brain, together with Ulf Siemens group in Tübingen and Gianluca Romani's group in Chieti, Italy. So we want to do EEG in closed loop with TMS. And in my opinion, fortunately, the biggest problem is not auditory or somatosensory artifacts. It's the targeting artifacts. So, but for that, we do need reliable EEG signals. So we're going to concentrate on the EEG artifacts in this lecture. A little bit about scientific critique. It can be annoying when we hear this, but it is essential for progress. So therefore, the critique is really welcome. And Hartwig started this in this context, so we really welcome that. Many of you may know this paper by Ilmoniemi in 2005. Why most published research findings are false. It's not only TMS EEG, but I don't know if that's true for this field, but you have to be really careful to make sure that the conclusions you make in your paper are based on rigorous logic from your experiments and from your data. I slightly disagree with Hartwig. You don't always need the control experiment. Sometimes you can come to the conclusion without a control experiment. Sometimes good control experiments are extremely hard to do. But you have to be able to just justify the conclusion based on your data and the data analysis. We have been studying TMS EEG since the mid-90s in Helsinki. A lot of papers, approximately 10 theses, have been published, many of them about the methodology. In this picture, one of my former students, Hanna Mäki, is stimulating the brain with a navigated TMS, and there's a 60-channel EEG device that has been used. So this started in 96. Here's our first small number of electrodes, first study. We moved the electrodes to different locations. And on the left, you see an animation based on these EEG recordings, slowed down by a factor of about 60. So the left hemisphere was stimulated at the cross. The coloring, the animation shows the estimated electrical currents in the brain, neuronal currents, as a function of time after the pulse. So you see that the left side is first activated, and then about 20 milliseconds later, there's activity on the right side. Hartwig showed the upper line of this picture, and that's a good idea to think about. Maybe that's not real neuronal activity on the right, but if you, and it might be an artifact. I'm open to criticism. But look at this when we stimulate the occipital cortex. Then it's not the sensory cortex that is activated on the right side. It's also the occipital cortex. So at least in one of these cases, I think the conclusion is correct, that the neuronal activity due to activation of the left hemisphere propagates to the right side. And we know that there are causal connections, and the timing makes also sense in this case. And no control experiments here. A few years later, 8 years later, Ilmoniemi et al. In Wisconsin studied subjects in wakefulness and during sleep. Exactly the same stimuli, same location. In wakefulness, there was a complicated response. During sleep, deep sleep, a simpler one. Again, no control study, but this appears also to be, the conclusion appears to be right, that during deep sleep, the connections in the brain are not as strong as during wakefulness. Now I go to the artifacts, which we really have to pay more attention to than we have done in the past in the community. So in the coil over the head, here's a figure of 8 coil that produces fairly focal stimulation in the brain. There's a very strong current flowing in. It can be 5 kiloamps, sometimes even more, and that gives rise to a magnetic field and the local activation in the brain, which then propagates along the neuronal fibers in the brain. The magnetic field is about 1 Tesla, and the electric field on the order of 100 volts per meter, depending on how you do it. So there's an electromagnetic artifact if you have the electrodes over the head at the same time. We want to measure microvolts, but in the electronics, TMS electronics, we are talking about kilovolts. There's also electrode polarization, and the electrodes may move. Eye movements and blinks cause artifacts. There's the auditory artifact, somatosensory artifact, as Hartwig pointed out. And there's also a muscle artifact. If you're stimulating, let's say, Broca's area, then the underlying, this muscle under the coil is also activated, and that produces an artifact. Now, how do you get rid of these artifacts? The electromagnetic artifact must be avoided by ignoring the signal during the pulse. The TMS pulse is extremely short, just a few hundred microseconds. In the electronics by Juha Virtanen in Helsinki, already in the 90s, in the first studies that we did, the input to the amplifier, later amplifier circuits, is blocked. There's a sample and hold circuit that blocks the input for about 2 milliseconds, and this way we avoid the electromagnetic artifact. Auditory artifacts can be avoided by using good ear protection, of course, also as silent coils as possible. And if you still hear, if the subject still hears the auditory click of the TMS, one can use masking noise. And one should also remember that the main component of the auditory artifact is 100 milliseconds, sometimes even longer. So the very early TMS EEG components are not affected. So in the example that I showed initially, we were looking at the first milliseconds. I'm quite sure that there was no auditory artifact there. Muscle artifact, this is what we have found the most problematic. It appears if the TMS is over muscles, for example, temporal muscle can be activated. It has a huge amplitude. It can be more than 1 millivolt, 1,000 microvolt. One of my students, Thomas Motanen, did a study and mapped the muscle artifact amplitude. It's largest where the muscles are. In the middle, over the midline, if you stimulate near the midline, even some part of the motor cortex, there's next to no muscle artifact. Now, sometimes we want to stimulate areas where we get the muscle artifact. And one method that turns out to be fairly successful is called signal space projection. Hanna Mäki included this paper in her PhD thesis a few years ago, projecting out muscle artifacts from TMS-evoked EEG. So the idea stems from the fact that from the idea, there's the idea that artifact topography is obtained from high-frequency signal over 100 hertz, which is free of brain activity. Most of the brain-evoked activity, TMS-evoked brain activity, is below 100 hertz, but there's strong high-frequency activity from the muscles. So if we high-pass filter, then we only see the artifact. And we can obtain the topography of the artifacts from these high frequencies, and then we can project it away. Here's a time frequency plot of TMS-evoked EEG. There's a huge artifact in the beginning, and extending up to several hundred hertz, and we know that the several hundred hertz is not coming from the brain, and then we look at the topography of these high frequencies. Here are the first three principal components in one subject plotted. The three principal components of the topographies elicited by TMS in the muscles. There's high frequency activity, and there are different kinds of topography superimposed, and these are the most significant ones. What we can do is that we can project these out, these muscle artifact topographies, also from the low frequency response. And we call this signal space projection. We can take 3, or we can take even 10 projections away, depending on the severity of the artifact. And each projection removes one dimension. If we have 60 channels, then if we remove 10 dimensions, we still have 50 dimensions in the signal. And this work was continued by Tuomas Mutanen in several papers, and now we call this technique source-informed reconstruction. On the left, you see a 60-channel EEG response to huge artifacts on the left, and then after removing signal space projection, we get rid of the artifact, but also some of the brain signal. So what we do is to form a minimum norm estimate in the brain based on the projected data, taking into account the projection, and then from that one can calculate the corrected, cleaned EEG, TMS EEG data on the right. It looks good, of course. Again, we have to be careful that maybe some of this is confounded by auditory or somatosensory artifacts. Another thing we can do is to estimate contamination levels. In the color figure in the middle is a contamination level map in different electrodes. How it is done is that you can estimate the signal in any EEG channel from the neighboring channels, actually from all the other channels, and because we have fairly low spatial frequencies in the EEG map, and then this can help one to optimize the cleaning of the EEG signals. And these techniques, again, due to Tuomas Mutanen, is called sound source space utilizing noise cancellation. So I explained a few methods to get rid of the artifacts. So final points here. First, try to avoid artifacts. The auditory artifact, use as silent coils as possible. Use good auditory masking, hearing protection, and then if there's still sound from the TMS pulse, masking. Try to avoid or minimize the muscle artifact. Well, then you still have artifacts. Try to separate the signal and non-signal components. Usually the artifacts and the brain signal have different topographies. In the signal space, they point in the different directions. Therefore, it's possible to use this SSP or signal space projection. But you need good data. Also, you also need to measure the artifact at a high quality to be able to use the projection method correctly. And then there's the SIR and SOUT algorithms. And Hartwig already mentioned independent component analysis. Many people, like we previously, also used it, but then I realized that indeed, as Hartwig already said, the TMS evoked signals and the artifacts are not independent at all. They are fully dependent. Dependence means that if you observe one signal, you can predict that there's the other one. So independent component does not produce reliable results. One of my students, Johanna Metsomaa, developed methods to deal with this, to make the muscle response and muscle artifact and brain response more independent. And finally, make sure that your conclusion is supported by the data. Often control experiments are not needed, in my opinion, but often they are needed, and then you should use them. Also, sometimes you don't need, if you want to find out phenomenon, you don't need many subjects necessarily. I want to acknowledge many people, many of my students and collaborators for this work, and funding agencies and universities that have supported this work over the years. And for more details on EEG and MEG signals, see our new book, Brain Signals. And thank you for your attention.